Hello my dear students, this is Dr. 𝗲𝗵𝗵𝗺, your biochemistry faculty. In this video, we are going to discuss about the structure of DNA and about the properties of DNA. So first of all, let's see DNA structure and properties. Now, when we talk about DNA, what is the full form? DNA stands for deoxyribonucleic acid. Deoxyribonucleic acid, which means that it is formed of three components. It has a base, it has sugar, which is attached to phosphate. So if three components are there, we will call it as a nucleotide. But if 2 points are there, means base is combined with a sugar, means no phosphate is there, we will call it as a nucleoside. Now coming over to this, if we talk about the sugar in DNA, the sugar in DNA is deoxyribose. Right? And deoxyribose means obviously it's a pentose sugar, but the second carbon atom has HH here. Right? Rest everything will be the same. So this represents deoxyribose sugar. Then if we talk about the bases are again of two types. The bases are purines and pyrimidines. Now if we talk about purines, purines are adenine and guanine, and pyrimidines are thymine, cytosine and uracil. But since uracil is present in RNA, here we are talking about DNA, so uracil will not be present, but since they are the three bases, and we all know that purines and pyrimidines are synthesized by a de novo pathway and a salvage pathway. Now the most important point which we have, because in our previous videos we have discussed already what is a de novo pathway, what is the salvage pathway, and how these purines or pyrimidines are synthesized, how they are catabolized, how they are utilized in the body. Now here what we have to remember is, if I talk about purines, they are actually forming AMP or GMP, right? So this AMP or GMP, or you can say CMP, means the sugar in them is ribose sugar. So this ribose sugar has to be converted into deoxyribose sugar, where we can say DAMP will be there, DGMP will be there, and DCMP will be there. So I can say ribose to deoxyribose sugar. So this type of reaction will be called as conversion of the ribonucleotides to deoxyribonucleotides, and how it is made possible. So this is a very important reaction, conversion of ribonucleotides to deoxyribonucleotides. So what happens here is, the reaction is reduction, right? And the enzyme here is ribonucleotide reductase, very important. So I can simply say that the reaction is AMP or GMP in the presence of thioredoxin reductase or ribonucleotide reductase is going to be converted into DAMP or DGMP. And ribonucleotide reductase consists of thioredoxin. So thioredoxin is there, reduced to thioredoxin oxidized. And this is coupled to NADPH plus H positive, like this. So we can say that this is how the DNA and RNA bases are synthesized. Now the most important point here to remember is, because this video is actually on DNA structure and properties. So first of all, if we talk about the properties here, DNA absorbs at UV light or UV range. This is very important. Absorbance of DNA. Yeah, DNA absorbs at UV wavelength, which is 260 nanometers. And this absorbance is due to the composition of the bases. This absorbance is due to the composition of the bases. Very important. Now coming over to this, if we talk about the bases only, I told you there are 4 bases: adenine, guanine, cytosine and thymine. So the 4 bases which are present in DNA are adenine, guanine, cytosine and thymine. Now the question is that what is the composition, because I have mentioned here about the composition of the bases, in what proportion they are present. So this composition was given by a scientist named 𝗏𝗏𝗃, and they were famous as the 𝗏𝗏𝗃 rules. So if we talk about the composition of DNA, it is given by 𝗏𝗏𝗃. And he said that amount of adenine is equal to amount of thymine, amount of guanine is equal to amount of cytosine, and amount of adenine plus guanine is equal to thymine plus cytosine, or amount of adenine plus guanine over thymine plus cytosine is equal to 1, which is constant for a given species. Ab we know adenine, purine, thymine, pyrimidine. Okay, so this is the composition, or we can say amount of purines is equal to amount of pyrimidines, and this is equal to 1, constant for a given species. Very important, right? So if we talk about these, and always remember one thing, that adenine and thymine are linked by double bonds, and that too hydrogen bonds, and guanine and cytosine are linked by triple bonds, but obviously hydrogen bonds. So the most important another point is that adenine is linked by thymine by a double bond, and guanine and cytosine are linked by triple bond. So obviously more energy will be required to break guanine and cytosine than to adenine and thymine, and this becomes the basis of differentiation of DNA. So DNA which is having more of GC base pairs will have more absorbance than AT base pairs. So I can say that DNA having GC base pairs has more absorbance than DNA having 80 base pairs. This is very important. So we can say that if the DNA is having more GC base pairs, that DNA will have more absorbance and we will call it as a hyperchromic DNA. And that DNA which has more 80 base pairs will have less absorbance and we will call it as hypochromic DNA. And how this absorbance is studied? It is studied when DNA is absorbing at 260 nanometers, which is the ultraviolet range. Then we come over to what is this is composition of DNA, what is the structure of DNA. So if we talk about the structure was given by Watson, Crick and Wilkins. Watson, Crick and Wilkins. They were the three scientists who gave the structure. Wilkins gave the X-ray or the three-dimensional structure of DNA. So whenever we talk about Watson and Crick, the most important, first of all, I'll just draw DNA is double-stranded like this. So first is DNA is double-stranded. The two strands are anti-parallel to one another. Anti-parallel. That means if one is running in the direction 5' to 3', the other will run in the direction 3' to 5'. So one runs in 3' to 5', the other runs in 5' to 3'. And Watson and Crick basically emphasized on the B-type DNA. This is also very important. Okay, so this is actually the B-type DNA. Then the diameter is 20 angstroms. There are 10 base pairs per turn. Right, so we can say 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10. And distance between successive base pair is 3.4 angstroms. At overall is 34 angstroms. Okay, so we can say two strands are anti-parallel and then the width is 20 angstroms or diameter. There are 10 base pairs per turn. There are 10 base pairs per turn. And distance between successive base pairs is 34 by 10 equals to 3.4 angstrom. And obviously it has a minor groove and major groove. It has minor and major grooves. Very important. So this is these are the postulates given by Watson and Crick, and this is basically about B-type DNA. Okay, the structure of, I can say, B-type DNA. Because, and it is obviously right-handed because there are 5 types of DNA. If we talk about, there are 5 types of DNA. Right, which is A, B, C, D. They all are right-handed and Z is left-handed. But in our body, mainly B type of DNA is there. So this is the structure as given by Watson, Crick and Wilkins. Wilkins was the one who showed the three-dimensional structure, and he gave this kind of this thing that there is a minor groove and a major groove when DNA is turning. Then after the structure, then we come over to the types of bond which are present. The type of bonds. Now, when we talk about the bonds, we know that DNA is double-stranded. So, it's like adenine with thymine, which is further attached to sugar, which is further attached to phosphate, like this. Guanine and cytosine, like this. Now, the question is, what is the bond present between the bases? What is the bond present between the base and the sugar, and what is the base bond present between the sugar and phosphate? So there are basically three types of bonds. So if we talk about the bonds between the bases, they are hydrogen bonds. Okay, then bonds between the base and sugar. It is beta N glycosidic bond. Right? And the bond between the sugar and phosphate is phosphodiester bond. So these are the three main bonds present. Phosphodiester bond. Right? So we can say bond between the bases, hydrogen bonds, bonds between the base and sugar, beta and glycosidic bond, and bond between sugar and phosphate, phosphodiester bond. So these are the three types of bonds which are present in the structure of DNA. Right. Then after this, then we come over to the next one. How this DNA is organized. Yeah, what is the organization of this DNA? Because ultimately, if we talk about DNA, it is actually the basic, I would say, which is bearing all the genetic information which is being transferred from the parents to the offsprings. Right. So then comes organization of DNA. Now, this DNA is organized with the basic proteins known as histones and non-histones. Very important. So, this DNA combines with histones and known as nucleoproteins. And if we talk about histones, what are histones? They are the proteins, yeah, basic proteins rich in lysine and arginine. Very, very important. These histones are actually the basic proteins which are rich in lysine and arginine, and they are of four main types. Four main types, which are H2A, H2B, H3 and H4. So they are arranged as an octamer. Means H2A, H2B, H3, H4. 4, 5, 6, 7, 8. And DNA is arranged like this. And this compact structure is known as a nucleosome. There is one more which is known as H1, and this H1 is known as a linker DNA. Right, and this nucleosome is as it is present in the on the chromosome. So I can say that if say this is the chromosome, this is the centromere, and these are the chromatids, and here this thread like structure is there. Here this structure is present like this, and one small H1 is present like this, which is also known as the spacer DNA, or it is also known as the linker DNA. Now this resembles like as if beads are there in the necklace. So that is why this is known as a beads on a string appearance. Beads on a string appearance. Very important. So basically the nucleosomes are present in the form of beads, and this is H1 or the linker DNA. So these histones play a very important role. Whenever DNA has to perform some function, it will be released from histones. But whenever DNA is not performing any function, it will be enclosed with the histones. And that is why we say that the function of these histones is it plays a very important role in the gene expression as well as the regulation. Okay. So this is what is the organization of DNA with histones, and on the basis of this we can say that there are two types of chromatin fibers also. And what are these? One is euchromatin, and heterochromatin. So if we talk about the differences between them, U mane new, or euchromatin is an area which is less stained or light stained area. Right, and heterochromatin is darkly stained area. Right. Then if we talk about euchromatin, it is transcriptionally active. So it is transcriptionally active, and this is transcriptionally inactive. This is having permissive chromatin, and this is having repressive chromatin. And chromatin is just that mesh-like fibers which is present in the chromosomes, which is carrying this DNA from the parents to the offspring, which is actually having the entire genetic material. So this is just the organization of DNA. Then another important point is, जैसे I told you कि DNA is double-stranded. In some viruses and all, DNA is single-stranded also, right? But there won't be any hydrogen bond between the bases because it is single-stranded. Double-stranded है, so the two strands are complementary also to one another. Means adenine will always base pair with thymine with double bond. Guanine will always pair up with cytosine with a triple bond. That is a mandate. And the two strands are anti-parallel to one another. Means if one is running in the direction 5' to 3', the other is going to run in the direction 3' to 5'. Now after organization, then we come over to that this DNA is basically can undergo denaturation, it can undergo renaturation, right? Or you can say we can easily denature the DNA strands, and this at the temperature at which this denaturation of DNA is occurring, we call it as the melting temperature of DNA, which is also known as Tm. So what is Tm? It is the melting temperature of DNA. Or how we define this temperature? It is the temperature defined as the temperature when half of the DNA strand is denatured. Is denatured. Very important. So I can simply say that DNA having GC base pairs has more Tm than DNA having AT base pairs. Very important. So DNA having GC base pairs will have more Tm than DNA having AT base pairs. Why? Because GC has triple bond, AT has only double bond. So this is again very, very important. Then after Tm, then we come over to denaturation. Now if we talk about denaturation, simply you can say a removal of the naturally existing state of DNA. Denature. So how do we define denature? You can say removal of the naturally existing structure. Now it can be due to so many factors. Right, by you can say change in temperature, right, whether it is increased, whether it is decreased, by addition of alkalis or by addition of acids. Similarly, formamide also destabilizes the bonds. Formamide also destabilize the bonds. And here always remember, the hydrogen bonds are broken. Not the phosphodiester bond, not the beta and glycosidic bond, only the hydrogen bonds will be broken. So whenever we are increasing the temperature, the bonds will break because hydrogen bond is more susceptible towards the heat. So what we do is we try to bring the temperature down, thereby reuniting the DNA strands. That process is known as annealing, leading to renaturation, right? So there are two terminologies related to denaturation only. De means removal, then renaturation. What is this renaturation? Reunion of the strand by joining the bonds, right? So slow cooling of DNA is annealing. Very, very important, because most of the students get confused between these three terminologies. So always remember, denaturation means a removal, or you can say breakdown of the strands, whenever we increase the temperature. So we have to join the strands, jaise in RFLP technique, in different hybridization techniques, renaturation and denaturation are the main pillars. So when we try to bring the DNA strand together, it cannot be brought immediately together, so slow cooling of DNA is done. So slow cooling is annealing, which will lead to the formation of renatured DNA. So these are the three very important properties. And always remember, DNA is double-stranded. The composition of DNA was given by 𝗏𝗏. Just suppose if I asked you that if any sample of DNA is there and if adenine is 23%, right, what will be the concentration of guanine? So these type of numericals can be asked. So if adenine is, so what will be the solution to this? That if adenine is 23%, I told you that adenine is equal to thymine. So thymine is also going to be 23. So overall it will be 646. And then guanine we have to find out. So guanine will be equivalent to 100 minus 46. So it comes out to be 6,410, 448, 554, sorry, 54910 like this. So 54 divided by 2. So it will be equivalent to 27. So the answer will be guanine will be equivalent to 27%. So these are the questions which have been asked in the exam so many times. Ki composition is given, of one base is given, you have to find out the other base. Okay, so I told you, you just have to follow the 𝗏𝗏 rules. Then this DNA, once it is produced, it is undergoing a central dogma of information, which means that this DNA can be converted into RNA. And this RNA is going to translate the information in the form of proteins. And this DNA can back be converted into DNA. So this process is known as replication. Formation of RNA from DNA is known as transcription. And formation of proteins or translation is known as translation. And this RNA can also be converted into DNA, which is a reverse of transcription. They say in COVID it was there. Or you can say majority of the retroviruses, HIV virus, their reverse transcription is there. Right. So we call this as a central dogma of flow of information. And this is basically bidirectional after the discovery of the AIDS virus or retroviruses. So we can say DNA will let the information flow to form RNA, and this RNA will translate the information in the form of proteins. Very, very important, right? So this video was exclusively about the DNA structure and organization. If we talk about the structure, always keep one thing in mind, the Watson and Crick model, and you should know the type of bonds present. If we talk about organization, you should know what are histones. Because whenever we are going to study about gene regulation and expression, we are going to study this histones in detail. Because it is like a nucleosome which is present on the chromonemator or thread-like structure. So it is inherited like this. So that's all about this video. I hope you have understood about the structure and organization of DNA in detail. Thank you so much.